Despite recent advances in rheumatoid arthritis therapy, there is still a need for alternative treatments. Orencia represents a therapeutic approach to rheumatoid arthritis with a fundamentally different mechanism of action to current treatment strategies. It is the first FDA-approved agent to selectively target a key co-stimulatory signal required for full T-cell activation. Rheumatoid arthritis is a complex autoimmune disease. Recent models of rheumatoid arthritis pathogenesis show that multiple pathways and cell types are involved. In the synovium, activated immune and non-immune cells participate in the inflammatory cascade. Macrophages, T cells, and B cells release cytokines, autoantibodies, and other inflammatory mediators. These mediators stimulate downstream effector cells, resulting in further inflammatory mediator release from chondrocytes and osteoclasts, and causing fibroblast-like cells and other cells to directly attack joint structures. Together, these processes promote joint inflammation and destruction. These immune processes are orchestrated by the activated T cell. The important role of activated T cells as orchestrators of rheumatoid arthritis makes T cell activation a rational therapeutic target. In the normal immune response, naive T cell activation occurs through the following steps. First, antigen is processed and presented to the T cell by the antigen-presenting cell, or APC. Although several possible antigens have been suggested as pathogenic triggers in rheumatoid arthritis, a specific antigen has yet to be identified. The antigen is then recognized by the T cell through interaction with the T cell receptor, or TCR. This is signal 1. Following this, a co-stimulatory signal, or signal 2, is required to promote full T cell activation. There are several co-stimulatory pathways. Signals through these pathways can either upregulate or downregulate T cell activation. A key co-stimulatory pathway is the engagement of CD8086 on APCs with CD28 on T cells. This produces positive co-stimulatory signals and facilitates full T cell activation, proliferation, survival, and cytokine production. CTLA-4 expression is upregulated 24 to 48 hours after T cell activation. It has a much higher binding avidity for CD8086 than CD28 does. In the normal immune response, CTLA-4 competes for CD28 binding to CD8086 and produces a negative signal, causing downregulation of T cell activation. Orencia is a fully human immunoglobulin-soluble receptor fusion protein, comprising the extracellular domain of CTLA-4 and a modified fragment of the Fc domain of IgG1. Orencia acts by binding to CD8086 on APCs and preventing its interaction with CD28 on T cells. The unique upstream action of Orencia at the level of the T cell leads to selective inhibition of T cell activation, as well as the downstream activation of effector cells and the subsequent release of the cytokines and inflammatory mediators that cause the inflammation and destruction characteristic of rheumatoid arthritis. By selectively modulating the CD8086-CD28 pathway, Orencia is expected to allow other pathways to remain largely intact, such that T-cell activation is not expected to be completely blocked. Other pathways which modulate T-cell activation may continue to function. In clinical trials, Orencia at a dose approximating 10 milligrams per kilogram decreases levels of inflammatory mediators such as IL-6 and TNF-alpha, which are released by the multiple cell types involved in the inflammatory cascade. Orencia also reduces rheumatoid factor release and levels of C-reactive protein. Levels of soluble IL-2 receptor, a membrane-soluble receptor that is synthesized by activated T cells, and of matrix metalloproteinase, MMP3, a mediator of joint destruction, are also decreased. The relationship of these biological response markers to the mechanisms by which Orencia exerts its effects in rheumatoid arthritis is unknown. Orencia has a fundamentally different mechanism of action than other currently available rheumatoid arthritis treatments.